Hey, so we are back with the third lecture of chapter 12, and in this lecture we're going to talk specifically about the steps, or the intermediate steps of DNA replication. Now we left with this slide, and we were talking in length about the central dogma. We talked about how DNA becomes RNA, how RNA becomes proteins, and that DNA becoming DNA, or DNA copying itself, is called replication, and that happens in the nucleus. DNA making RNA, specifically mRNA, is called transcription, and that too happens in the nucleus, again because we want to keep DNA in the nucleus at all times. Once RNA is made in the nucleus and introns are cut out and exons are spliced together, we will take that mRNA message out into the cytoplasm, where we will meet up with a ribosome and produce a protein, and that's called translation. Okay, so we're going to go in depth about this blue process right here. In further lectures we'll talk about the RNA transcription and then protein translation. But for now, let's talk about what DNA replication is and how DNA is copied. We've talked about the cell cycle, and we've talked about what happens in a cell before it can divide, and we talked in depth about the G1, S, and G2 phases. Remember, the organelles had to be copied, all the proteins had to be copied, and the cell had to grow to a more mature size in G1. When it gets to S, synthesis phase, the DNA has to copy itself. And why does the DNA have to copy itself? Well, it's because we need two copies of DNA in order to divide the cell into two new daughter cells. One daughter cell needs a copy of DNA. So if you're going to make two daughter cells, you need to have two copies of DNA. That division of DNA happens in S phase. Okay, we talked about that, synthesis phase. DNA is a double-stranded molecule, and so in order to copy DNA from the two strands, it has to be unzipped and unwound. And we're going to learn a lot of different enzymes in this particular lecture. And the first enzyme up is DNA helicase. Helicase is an enzyme that's going to break hydrogen bonds as it unzips and unwinds this DNA molecule. Okay, and again, it has to unzip and unwind this molecule because we have to get to the base pairs in order to use the two template strands as a recipe, so to speak, so that we can make the new strands from the template. And DNA can only be replicated in the presence of an enzyme. So we're going to learn a lot of different enzymes. We know when DNA is replicated, it's in S phase of the cell cycle, and we know that enzymes play a big role in anything biological, and DNA replication is definitely no exception. So these are a few of the enzymes that are in or responsible for DNA replication. We already talked about DNA helicase. Remember, helicase is the enzyme that's going to break hydrogen bonds as it unwinds and unzips the DNA double helix. RNA primase is an enzyme that's going to lay RNA primers every few thousand base pairs. Okay, there are usually between 5 and 10 RNA primers laid about every 2,000 base pairs. Okay, DNA ligase is an enzyme that will kind of weld together two smaller DNA fragments, or if there's a mutation that happened, or some kind of substitution in the proper DNA sequence, DNA polymerase can actually cut that out and replace with the proper base pairs, and then DNA ligase is kind of the Mr. Fix-It that comes in and welds any of those problems together. DNA polymerase, there's actually two, DNA polymerase 3 and DNA polymerase 1. We're going to talk about both. DNA polymerase 3 will come first, DNA polymerase 1 will come second. You'll remember of a situation similar when we talked about the photosystems, where photosystem 2 comes before photosystem 1. Okay, well, that kind of situation happens again. And then we have single-stranded binding proteins, which are going to play a huge role in actually holding the replication forks and the replication bubble open, so that the DNA strand doesn't wind back up on itself. So DNA is copied in a semiconservative fashion, meaning one parent double helix is going to open up, meaning these two strands are held together by hydrogen bonds. Okay, so G is a base pair with C, A is a base pair with T. That always happens. We're going to unzip and unwind this DNA molecule, and so we break the hydrogen bonds that are holding these G's and C's and T's and A's together. Once we open up this DNA double helix and we have both template strands basically separated, then we're going to use both template strands as a recipe or template in order to create the two new lighter blue strands based on the template strand. Okay, so these would be identical to the original strand here, and these new nucleotides would be identical to the template strand here. And by using both sides of the original parent strand, we were able to basically build two new daughter strands, which are identical to the original template strand. And now we have two identical double helices, which can be given one to each cell, and we've kept our genetic continuity throughout this whole process. Here's another way to look at it. So early on, there was believed to have been a conservative model or a conservative approach to DNA replication. Then they thought about this kind of semi-conservative model. They also thought about a dispersive model. Now, these three are ways, almost like theories, ways in which people over the years have described or tried to describe the way in which DNA is replicated. Conservative has been disproven because we know now through the measles-Stall experiment that DNA actually is made in a semi-conservative way. Okay, so DNA is made in a semi-conservative way. This is the correct way. This is wrong, and this is wrong. This is basically saying I have one parent strand. I'm going to look at this parent strand and I'm going to make a completely new strand based on what I think this looks like. They don't open it up, they don't unwind it, they don't break any bonds, they don't use anything as a template, and so they're just building a new strand based on what they think this looks like, and that would result in a lot more mutations than we currently have. Dispersive model is exactly what it sounds like. You're basically piecing the old and new strands together, and so you're saying this old strand can be chopped up like a chef chopping up an onion, and half of this old strand can be given to this new strand, and half of that old strand can be given to this new strand, and then you build the DNA strands between them, which makes absolutely no sense. And it makes no sense at all because you would have nothing to use as a template to build in these new gaps. Okay, if you have an old gap here and you have an old gap here, well, what are you going to use as a template to build in kind of this or bridge the two old sections of DNA? You'd have nothing to build that new strand off of, and so that's absolutely wrong as well. Semi-conservative is the way in which DNA is made. They open up, all cells in your body will open up the DNA molecule using helicase to expose both template strands. They will use this old strand as a template to make this new strand off of. They'll use the other old strand to make this new strand off of, and you will produce two new daughter strands that were made semi-conservatively. Again, semi-conserved because you are conserving one strand per DNA molecule. So this DNA molecule has one strand conserved from the original parent. This strand has one strand conserved from this parent. This strand has the original parent strand conserved as well, and so you'll even see throughout the second replication you still conserve one strand of DNA. A few facts about semi-conservative replication. DNA helicase, like I said, was the first enzyme we talked about. It's going to unwind the DNA molecule. It's going to open up the DNA strand by breaking hydrogen bonds. Hydrogen bonds are the weak bonds that are holding the complementary base pairs together. DNA polymerase is the enzyme, and this is specifically DNA polymerase 3. DNA polymerase 3 is going to read the bases on one side, that is the template strand, and lay down the new complementary bases on the new complementary strand in a 5' to 3' direction. Okay, so we're always going to build DNA from 5' to 3'. That means we're going to read the DNA template strand 3 to 5. Okay, and it makes sense. If we look at this DNA strand, this strand has to be 5 to 3. If we read the template strand going this way from 3 to 5, we will build this new strand 5 to 3. And remember, we always have to build DNA 5 to 3. Now, if we read it 3 to 5 and build it 5 to 3, then you will make the new strand anti-parallel with the original first. An old strand is used to make a new strand. We got that, right? That's semi-conservative replication. We're going to use an old strand, that is the template strand. We're going to make the new strand from that template strand. The DNA strand is copied from 5 to 3. We said that always happens. One strand is copied straight down in a continuous line. That is called the leading strand. The other strand is copied in the opposite direction, and that one will be made in fragments called Okazaki fragments. And we'll see pictures of that in a minute. DNA polymerase then proofreads the new strand to detect incorrect bases. This would be DNA polymerase 1. remember there was 2. DNA polymerase 3 comes first, DNA polymerase 1 comes second. Remember, DNA polymerase is the proofreader, which means that tells you exactly when it comes. It comes second. You don't proofread first, you have to write first. You proofread second. In the case of incorrect base insertion, meaning we put the wrong bases down, when we should have had an A, we put a G, or when we should have had a T, we put a C. DNA ligase will remove the incorrect bases. That's one of our enzymes that's going to come in. DNA polymerase 1 will then place in the correct bases, and then DNA ligase will come in, like I said before on that previous slide. It comes in and actually like welds the two kind of fragments together. Single strand binding proteins will keep replication bubble open, and they are paramount or really important when we're talking about holding the DNA replication bubbles open and keeping the replication forks open so that the double helix doesn't wind back up. Okay, if the DNA double helix winds back up, replication cannot happen. Okay, the replication bubble has to be open. The replication forks have to be way open in order for DNA replication to happen, because there's a lot of enzymes that have to get in there. If the enzymes can't get in, DNA replication can't happen. So if we move into a picture, this shows you the DNA model. Okay, this is a DNA molecule. It's a double helix. The DNA replication would be happening in this direction, in this particular image. Notice that you have the original blue strands. You'll see a blue strand here, and you'll see a blue strand here, but we also see these new kind of peachy pink or purple strands. This is a new strand, and it's being built this way, and then this strand is being built this way. Okay, and that'll make more sense here in a minute. But this is the replication fork, and the replication fork is the point that the replication bubble has kind of just opened up, or the base pairs that have just broken their hydrogen bonds. That's the point at that we call the replication fork. Okay, so let's talk about the semiconservative replication in a little more detail. Notice here's the 5' end of this strand. Here's the 3'. So this strand is moving that way, and this strand, the other original strand, is moving 5' to 3' in this opposite direction. So these two blue strands are semi-conservative, or rather, they are anti-parallel. Those are the parental strands, the template strands. Because this top template strand is running 5' to 3' from left to right, we have to build the daughter strand from right to left, because remember, we have to read the template strand 3 to 5 while we build the new DNA molecule 5 to 3. So we're going to read it from right to left, and we're going to build it from right to left. And like we said before, DNA polymerase 3 is the enzyme that is going to build this new DNA molecule. Over here, same enzyme, DNA polymerase 3 is going to come in, and it's going to actually build fragments. And the reason it has to build fragments is because notice this template strand is running 5' to 3' from right to left. And so we have to build our new molecule from left to right. But we can't build it left to right because our replication bubble is moving from right to left. Okay, and so as this replication bubble opens up, we literally have to build these in fragments. So we have to build this one, and then we have to jump ahead to where the replication bubble opens, and we build it back to where we came from, and then we jump ahead and we build it back to where we came from, and then we jump ahead, and we just got to the replication bubble, so now we're building it back to where we came from. But by the time we get to ahead, or the replication bubble, it would have opened up more, and so you can see how many smaller fragments we had to make in this left to right direction. We call those Okazaki fragments. And we call this the lagging strand. Okay, this strand is down here is called the lagging strand because we have to make it in a non-continuous motion. This is called the leading strand because we can make it in a continuous motion. Remember, we said DNA ligase is the enzyme that comes in and will actually weld these fragments together. So DNA ligase will weld this together, then skip ahead, weld that together, skip ahead, weld it together, and so when we're done, we'll have a continuously made strand, but we would have made it in smaller fragments. This shows you the same thing. So when you open up a section of DNA, you always have an origin of replication, and it runs right through the middle. The replication bubble will open from right to left and left to right from that origin of replication. So we're going to be moving in both directions, just like you were to unzip a Ziploc bag. Okay? when you unzip a Ziploc bag, you're actually going to pull it from the middle, and it's going to unzip equally in both directions. And so here we will start at the primers that are laid at this origin of replication. That's what these red primers are showing you. So RNA primase comes in, lays primers here, usually 5 to 10. Then DNA polymerase III comes in and lays the DNA molecules, or the DNA nucleotides, in a continuous 5' to 3' motion. This is the lagging or the leading strand because it's continuous. Down here, we're going to lay our RNA primers, and then DNA polymerase III is going to come in and lay DNA nucleotides in a 5' to 3' direction. Okay, so the top left of this bubble and the bottom right are both leading strands. The bottom left and the top right are going to be lagging strands. Okay, and so why are they lagging strands? Well, our replication is going to be happening from right to left as this replication bubble opens up. And as this continuous strand is being made, RNA primase is going to lay a few RNA primers and then skip ahead and lay a few primers, and then skip ahead and lay a few primers. And then DNA polymerase III comes in and lays the DNA. Okay, if I change colors, might make it a little easier to see. Okay, come in here with blue. So RNA primase comes in, lays the primers, and then DNA polymerase III comes in and lays the DNA nucleotides. And then they come in and lays the DNA nucleotides. And then it skips ahead, lays DNA nucleotides between the primers. Okay, what are these fragments called? These are called Okazaki fragments. Then DNA polymerase I will come in and will pull these primers out, and will replace the primers with DNA nucleotides. And then once they replace them with DNA nucleotides, then ligase comes in and, like I said before, will stitch these fragments together, or weld the fragments together into a continuous strand. This is probably the best way to look at it. It shows you individual steps. It shows you the individual nucleotides. And so if we look at the edge of this page, okay, the far right edge of the page, you will see basically the origin of replication. Notice this box is right here, and the far right, right here on this page, is the origin of replication. So just off the screen is the origin of replication. RNA polymerase would have come in and would have laid RNA primers at that origin, and then DNA polymerase comes in and makes this continuous strand. This is DNA polymerase III, makes the continuous strand of DNA from a 5' to a 3' direction, because the template strand is 5' to 3' from left to right. Notice these single strand binding proteins, they are holding this replication fork open, and this is the replication fork. This is helicase. Helicase is like the zipper on the track, or the zipper track. It's moving down the train track, so to speak, this way, unzipping the DNA molecule by breaking hydrogen bonds. And so that leading strand is made very easily, right? You just lay your RNA primers, and then you lay in a 5' to 3' direction the DNA nucleotides to produce that DNA strand. Down here on the lagging strand is where it gets a little more confusing. All right, the first thing that has to happen is the RNA primase has to come in and lay RNA primers. Then it jumps ahead, lays RNA primers. Then it jumps ahead, lays RNA primers. Then it jumps ahead, RNA primers. Jumps ahead, RNA primers. And so it just leapfrogs down the lagging strand. And the reason we need RNA primers is because the DNA polymerase III is an enzyme that really doesn't have direction in life, and so it needs a starting and ending spot. Okay, and so once you've placed these primers, you will take DNA polymerase III and you will fill in the gap between primers, and then it'll jump ahead and it'll fill in the gap between primers. Okay, now I know it already did it down here, so we're kind of jumping ahead. Once DNA polymerase III fills in the gap, DNA polymerase I will come in and replace the RNA primers with DNA nucleotides. Once it places the DNA nucleotides in the place of the RNA primers, then DNA ligase comes in, and see this little gap right here? It's going to weld that gap shut, or weld these two fragments together, and then we get a continuous strand between two smaller individual Okazaki fragments. Once all of these primers are placed, then DNA ligase will come in and weld that hole shut. And then it'll jump ahead and weld this hole shut. And then jump ahead and weld that hole shut. Okay, and so it's an assembly line of protein and enzyme interactions as the leading and lagging strands are produced. Okay, and so all of these enzyme-mediated reactions that happen kind of like one after another, after another, after another, are just kind of the small stepping stones in the replication of DNA, and that happens in every cell in your body all the time. Little information just to kind of finish this one up. So if you accidentally put in the wrong base pairs, notice they didn't bond very well, the wrong base pairs are put in, nuclease enzymes, or enzymes that digest or break down nucleic acids, will come in and will pull out the wrong base pairs. Okay, notice it's pulling those out. And then DNA polymerase 1 will come in and fill in the gap with the proper DNA base pairs before DNA ligase comes in and actually bonds the two fragments together. This shows you just kind of a zoomed out version of semiconservative replication. One of the key things to keep in mind is that on one long strand of DNA, there isn't one replication bubble. There is a bunch of them. And so if this is the strand and we open it up 3 times, you're going to be 3 times as quick in your synthesis or in your replication of this DNA molecule. If we only opened up 1, it would take 3 times as long. In reality, one chromosome or one strand of DNA is going to be opened up multiple times. There might be 100, there might be 1,000 origins of replication that are happening simultaneously. And so all of these origin of replications that open up are just increasing the efficiency of that cell replicating the DNA, because we want to be efficient. We want to replicate our DNA quick. That's going to do it for semiconservative replication. It's going to be important for you to bring questions that might have arisen as you watch this lecture to class. We're going to watch videos in class and hopefully kind of make this make more sense. Until then, see ya.